cloning. Imagine, just for a moment, that we possess the delicate ability to rewrite the very instructions of life. We'll go step by step, reading the pages of this biological narrative slowly and carefully, understanding how scientists manipulate the microscopic world with profound grace.
Molecular cloning is a foundational technique in biotechnology. It's really not just a routine or sterile laboratory task. Rather, think of it as a continuous, precise story of assembling and rewriting a living blueprint. We use highly standardized enzymatic manipulations to direct the replication of recombinant DNA molecules within a host organism. We are, in essence, becoming authors of a microscopic text.
To begin our journey, we must first understand our vessel, which is the plasmid. A plasmid is a circular destination vector. You can think of it as a secure, looping vehicle that will carry our precious target DNA sequence safely into a new living home. It's the chariot that will transport our genetic protagonist across the boundaries of a single cell.
Preparing the DNA blueprint, isolating the hero. Like any great piece of literature, our narrative begins by isolating our hero, the gene of interest. We must gently and accurately separate it from its vast original environment before it can begin its journey. This preparation unfolds in two careful steps. First, we have gene isolation, which is commonly achieved through a process called polymerase chain reaction, or PCR. Following this initial isolation, we gracefully move to the second step, known as the restriction digest. Here, we carefully prepare both our isolated target insert and our circular vehicle so they can be moved and joined together in harmony.
Now, to safely extract the target insert with immense care, we rely on high-fidelity DNA polymerases. These are very special enzymes that flawlessly copy the target sequence. They ensure that the integrity of the sequence is perfectly maintained, without a single error in the genetic letters. Often, the flanking regions of this sequence are engineered to contain specific markers to guide its integration later on in our story.
Once our sequence is perfectly copied and isolated, we must prepare it to fit into the plasmid vehicle. We do this using restriction endonucleases. You can picture these functioning like delicate molecular scissors. With soft reverence, these enzymes recognize very specific palindromic sequences in the DNA. When they find these exact matching patterns, they carefully cleave or cut the phosphodiester backbone of both the genetic insert and the destination plasmid.
When these molecular scissors cut the DNA, they leave behind one of two beautiful types of ends. They can create sticky ends, which are slightly overhanging sequences reaching out to find a matching partner, or they can create blunt ends, which are perfectly straight, flat cuts. Both methods are distinct, yet both purposefully prepare the DNA to be perfectly joined together in our next chapter.
Building the recombinant plasmid, sealing the bond. With our microscopic pieces perfectly prepared, the separate pieces of our living blueprint will now be permanently united.
This union happens in two distinct and elegant steps. Step 1 is ligation, which is the physical joining of the DNA fragments. Step 2 is transformation, the delicate process of ensuring their safe entry into a completely new biological host organism.
Our builder for this joining process is an enzyme known as T4-DNA ligase. It quietly and efficiently catalyzes the formation of a phosphodiester bond. It smoothly seals the 3' hydroxyl end of one nucleotide to the 5' phosphate end of another. Through this quiet work, it effectively seals the DNA fragments into one complete, circularized recombinant plasmid. Once the plasmid is whole, it must find its new home inside a host cell, commonly a bacteria such as E. coli. But the cell membrane is a formidable protective wall. So, through methods like heat shock or electroporation, we gently alter the permeability of that bacterial cell membrane. We basically encourage the host cell to open its microscopic gates, allowing the exogenous DNA to safely leave the outside world and enter the protective cytoplasm.
Selecting the successful cells, finding the survivors. For the final phase of our narrative, we must discover which cells successfully accepted our genetic gift.
To find them, we quietly observe two final steps. First is selective plating, where we place the cells in a very specific environment. Second is colony verification, where we closely examine those resilient cells that thrive and grow into visible colonies.
The elegance of this design lies in our selective media. We plate the cells in an environment containing specific antibiotics. Our new recombinant plasmid carries a special shield within its sequence, an antibiotic resistance gene. Because of this, only the cells that have successfully acquired our newly built plasmid will survive the antibiotics to form colonies. All others, lacking the shield, will quietly fade away. To be absolutely certain of our success, we must verify the surviving colonies. We have several detailed methods to confirm that our newly placed DNA is correct in both its sequence and its orientation. We might use colony PCR to amplify the region, perform a detailed restriction analysis, or carefully read the exact genetic letters using Sanger sequencing.
Of course, a journey of this magnitude requires careful planning from the very beginning. There are highly detailed rules and critical considerations that guide this delicate scientific process to success. Let's gently review the boundaries that ensure our precision.
First, we must ensure enzyme compatibility. The chosen restriction sites must not occur internally within our gene of interest, or we risk tragically fragmenting our precious hero during digestion. Second, we utilize vector dephosphorylation. By treating the linearized vector with alkaline phosphatase, we remove the 5' phosphate groups. This gracefully prevents the empty plasmid from simply closing back up on its own, without accepting our target insert.
Furthermore, we must maximize competency efficiency, which we measure in colony-forming units per microgram of DNA. High-efficiency competent cells are absolutely essential when the ligation is difficult or our library of blueprints is highly complex. Finally, we must ensure orientation control through directional cloning. By using two different molecular scissors, we guarantee our DNA is accepted and positioned exactly right, preventing the insert from ever ligating in reverse.